Hello, welcome to the new lecture. Today I will be talking about the T cells and the different population of the T cell. So first of all, we have two type of T cell. Two type, yes, two type. Um, according to the receptor, we will divide them to the alpha beta T cell, gamma delta T cell. Okay, so when you go to the structure of the T cell receptor, there is chains alpha and beta, and the other is gamma and delta. So if you have alpha and beta chain, you will get this T cell. Okay, now 95% of the blood T cell will be alpha beta, and only small proportion, which is 10% to 5, is gamma delta. So this is the principal one. So as you know, according to the macrophage presentation, um, what will happen basically is that if this was your macrophage or any antigen presenting cell, the antigen presenting cell usually will give this antigen, present the antigen to the cell, which is the T cell, right? Naive T cell. Why it's a naive T cell? It's because it doesn't encounter any of the pathogen. Anyway, so it's come out from the thymus and the lymphoid organs. So the pathogen, the antigen presenting cell will encounter the pathogen in the blood or in the tissue, and then finally it will internalize it and give some of the particle on the MHC 1 or 2. So if it is presenting the particle in MHC 2, it will come in contact with a naive T cell. So this naive T cell will give the CD4, okay? But let's say the antigen presenting cell will present it on MHC1. If it is in MHC1, in this case, the naive T cell will become CD8, which is here, CD8, okay? So this is the first, oh my god, sorry, here, CD8. So this is the first principle. So accordingly, if it is MHC 1, it will be CD8. MHC2, it will be CD4. So then the macrophage will secrete some cytokines, and then this will lead to the differentiation to CD4. Now CD4 will decide which type of cell will give according to the cytokines it has. So let's say the CD4 has been secreting interleukin 4 and interleukin 33. Okay, interleukin 4, interleukin 33. In this case, we will give this Th2, which is the T helper 2 cell that is encounter the antigen via MHC2. The CD4 will secrete cytokines, and thus the lead. So this cell is important because it will secrete interleukin 4, 5, and 10. Now, interleukin 4 and 5 and 10 mainly responsible for the B cell stimulation, okay, and it will inhibit the formation of Th1, and it will stimulate the formation and proliferation of Th2, okay? That's it. This is all about the cytokines secreted via the Th2. We have also some of the stimulation of specific immunoglobulin like IgE and IgA. So according to the cytokines it secretes. Then CD4, if let's say the CD4 secretes interleukin 12, tumor necrosis factor alpha, and interleukin 18. In this case, you will get the Th1. Th1 is very important because it will secrete interleukin 2, interferon gamma. It will stimulate, this is interesting, it will stimulate the macrophage. So the macrophage that presents this pathogen will be able to phagocyte efficiently because of the help of the cytokines secreted by Th1. It also stimulates CD8, which is here, okay? So if you don't have Th1, you will not help CD8. CD8 will be weakly perform its function, okay? So it needs to enhance the function of CD8. And lastly, you have Treg. So Treg, it's T regulatory or suppressor T cell, which is responsible for the autoimmunity. Then you have Th17. Th17 is also important in the formation of autoimmune diseases and other things, and the inflammation. Okay, so if CD4 has been secreting interleukin 21, transforming growth factor beta, in this case you will have Th17 that will secrete interleukin 17, interleukin 22. So if CD4 has been secreting transforming growth factor beta, remember that you will have two cells. Why? Because this factor will stimulate Th9 and Th17. If Th2 has been secreting interleukin 4, it will stimulate the formation of Th9. Why? Because of the interleukin 4. Okay, so Treg, I think Treg, you have to. I forgot to mention that Treg is the one which basically will secrete. I'm sorry for this. Yeah, it will secrete interleukin 10, and it will secrete transforming growth factor beta. Okay, so transforming growth factor beta. So now you will be able to understand that, let's say the CD4 has been stimulated to produce this Treg. The Treg will produce for us the transforming growth factor beta that will also lead to the formation of Th9, Th17 because of this. And interleukin 10 that is secreted via the Treg is also secreted via the Th2. Okay. This is all about now let's talk about the function of them. Now the function of CD4 is to help or induce the immune response. Okay, so Th1 is the one which is which is important for antiviral and bacterial immunity. Why? Because it will stimulate the CD8 that is basically will kill the viral, right? So it is antiviral. And bacterial, why? Because it will stimulate the macrophage. The macrophage will lead to intracellular phagocytosis of bacteria like Mycobacterium tuberculosis. Okay, Th2 function. Th2 is immunity for extracellular parasites and allergic response. I mentioned this because it's the one which will enhance the formation of IgE. IgE is important for allergic and parasite formation, immunity. Th17, interestingly, is important for fungal and for inflammation. Okay, and for the CD8, it's important for antiviral and tumor surveillance. It's also important for some bacterial infection. And this is all about the population of T cell. Now this is what I've mentioned. If you have the, let's say here, the antigen presenting cell, according to the MHC, it will present either MHC class 1, I mean the surface, or MHC class 2. Now what happened here is that you have intracellular infection. So this is a cell in your body. Okay, this cell has a viral infection. It will present it on MHC1. Why? Because MHC1 is the receptor found in every cell in our body. It will give the antigen presenting cell, and then, or let's say, or I will not consider this because by itself, this cell can stem because it has MHC1, so it can immediately stimulate CD8. So this is, I'm not sure about this, okay, but I know that it has MHC1. But let's take this principle better. So the cell that is virally infected, it will present the viral on MHC1, and then that will lead to the presentation to CD8. CD8 will kill it by perforating granulocyte. Now this is cytosol-driven antigen because it is infected cell. Now in this case, this is the one that it's better to say about it. This is this is the antigen presenting cell. Antigen presenting cell has MHC2. This is the common route of presentation. It will internalize the extracellular peptides, and then it will form the vesicle, and then it will present it to the CD4. It has to be peptide. Okay, doesn't has to be peptide. Now let's say this viral, okay, doesn't present because some of the virus will suppress the MHC1. So how it will know that it has a virus. Now natural killer cell will perform the function. It will come and kill the cell that is not presenting MHC1. Okay. And this is the interaction between the, I'm sorry for this, there's a sound, yeah. Interaction between CD4 and antigen presenting cell. So basically what is happening is that the CD4, when it interacts with the antigen presenting cell, it has TCR. which is the T cell receptor. T cell receptor is important for the interaction with MHC class 2. Alright, this is the first thing. Second, TCR, now this is the receptor that is not working unless you have CD3. Why? Because TCR, the receptor for it is very short cytoplasmic tail. So how it can stimulate the intracellular signaling, right? It has to go to the nucleus and produce these cytokines, interleukin 2 and interferon gamma. So this CD3 will help phosphorylate some signaling molecules and lead to the formation. Now, LFA1 and ICAM, these are adhesion molecule. CD4 has to come like, okay, so imagine they are giving a present and someone giving you a present. You have to shake hand, right? CD4 do this. CD4 said, okay, I have to shake my hand with MHC class 2 in B cell region of MHC class 2. CD, because MHC has many regions. CD28 is another molecule, has to be contracted P7, and then CD4 ligand with CD4. So this is CD40. This is only like something that can stabilize the interaction between the cells. Okay, and this is all about it, and we will talk about in the rest of it in another model. But one of the slide, I think it's better to mention it here, that we should know that we have two types of, according to the function, T cell is divided into effector and regulatory function. If you take the effector function, you will have CD8 and CD4. Effector means that the cell will perform the function. So CD8 is the one which kills the viral infected cell and tumor cell, via granulosome and perforin. But CD4, they have many of the function. See, CD4 has two functions, regulatory or effector, but the CD4 that will lead to macrophage activation, which is basically Th1 via interleukin 2 and interferon gamma. In this case, it will lead to macrophage function, and then macrophage, when it's active, it will perform the phagocytosis. That's why it's effector function. Okay, it will lead to effect. Regulatory function, it will basically contact and cytokines. So CD4 will activate, as I mentioned, B cell. It will activate here, I mentioned to you, B cell. It will activate other T cells, here B cells and here the T cells, which is the CD8 cell. And lastly, regulatory response. Now this CD25 plus and CD4, I think it is surface molecule for Treg, okay, but I have to make sure whether it is Treg, but C17. But I think it is Treg, okay. And this is like a summary that CD4 will activate cytotoxic T lymphocyte, which is CD8, as I mentioned, from this pathway. CD4 will activate B lymphocytes, CD4 via Th2. CD4 will lead to macrophage activation via interferon gamma, and CD4 will lead to natural killer cell activation also, which perform the same function, but the difference between the cytotoxic T lymphocyte and natural killer cells is that the MHC presentation. If there is MHC presentation, the CTL will work. If there is no, natural killer cell will kill. So this is like a sister and brother. They will help each other, okay? Yeah, and that's it. This is all about it, and thank you.